Hi, Kevin Larry, mildly enthused to be here today with the gentlemen of Cofactor Genomics. They're a next-generation DNA sequencing company. And we're going to go ahead and meet those gentlemen right now. We have Reese, Jerry, and Mitch. And tell me, guys, what have you been up to lately?
So this is a really exciting time for Cofactor. Since we opened our doors last year, we've sequenced a large spectrum of genome projects ranging from bacteria, model organisms like the fruit fly, new organisms like the Syrian hamster, as well as human samples.
Oh yeah, that reminds me of a super boring time.
Exactly.
Mm-hmm.
Well, probably one of the more challenging projects we've worked on, both in the lab and computationally, has been the teddy bear genome project.
Yeah, that doesn't even sound remotely interesting.
So that's a project that we're very excited about, the teddy bear genome project, because it's an allooctoploid. So it makes it a very challenging genome to characterize.
So how does the process of doing all this boring stuff begin?
So first, the customer has to get us a DNA sample to sequence. We don't actually extract DNA from tissue or cells. We're a full services firm. We do handle everything from library construction, generating the data, and analyzing it. But we don't do the actual extractions.
Really?
There are, however, a wide variety of commercially available kits that we recommend. In fact, for the teddy bear,
QIAGEN, yeah, has a great kit.
Mm-hmm.
Has a wide variety of methods for extracting DNA from different kinds of teddy bears. Some are more difficult than others to get DNA from.
Assuming anyone still cares, why don't you guys show us the actual steps in this process?
So when a sample arrives, we put it through a number of QC measurements to judge its integrity. And if everything looks good, then we start the library construction process.
So this is typically how we receive our samples for sequencing. And really, you can never be too careful when handling somebody's samples. So you probably think I look a bit ridiculous, but this is how we make sure we're not cross-contaminating anybody's DNA.
So when do you think we're going to be done here?
Looks like they use the remains of the organism as hacking material. I guess I'll give you a couple minutes.
So once the library construction is complete, that's when the sample undergoes sequencing. And one of the most exciting things that we've seen is how quickly we can go from this quality data into actual publishable work.
So a couple questions for you guys. I thought it took years to build a good library, and you're saying you published a book in mere seconds?
That's really kind of the whole point.
Oh, yeah. Mm-hmm.
What?
Really boring. That's what that is.
You just take a little of this solution here, add it directly to the sample,
What time do you guys normally call it quits?
shake it up a little bit.
Hey, I know all of this stuff is really important and super boring, but I was wondering if I could go use the restroom.
And we're ready.
Looks like you could baste a serious turkey with that thing. Tell me about it.
Just load it directly into the mouse. And that's it. We should have results in about a minute or so. And there we go. A ready-made publication for Nature or Science.
So you guys are essentially living in a dream world, or a dream state, as I would call it.
Yeah, thanks.
And people are just running around with ponytails, and they're eating gumdrops, and going to the store every day, and buying really expensive shoes. I just don't understand how you guys can pull this off.
So not all researchers have been so quick to embrace next-generation sequencing, and they still do their projects with more classical methods.
So, Dr. Professor von Hasselhoff, what do you think about all of this next-gen sequencing mumbo-jumbo?
This next generation's just a fad. Just whippersnappers, that's all.
Yeah, it's all voodoo, right?
We used to do radioactive labeling, one at a time. That's all we did. This is just a fad anyway.
So, Professor von Handlebars, you seem to really have it together on this DNA junk, huh?
We used to take the gel, put it together, then we'd read off these letters, C, G, A, T. That's all we had to do.
So, Dr. Professor von Hasselstein, you must have sequenced a lot of DNA back in your day, huh?
Can we get this over with? I want to get home.
Professor von Stinklepants, I really like your style, especially when it comes to DNA sequencing. Maybe we should get dinner tonight. Say 2 a.m.?
Okay.